Hi, everybody. Welcome back. In the last couple of videos, we introduced the idea of protein design as an inverse problem of the protein structure prediction or protein folding problem, and then introduced both Monte Carlo and genetic algorithms as possible algorithms we can use to solve the kind of mixed discrete in sequence space and continuous in structure space optimization problem that protein design poses. Monte Carlo and genetic algorithms are both very powerful, widely used optimization algorithms. They're very general purpose. They work for almost any type of optimization problem you can imagine. You can frame them with, in the context of those algorithms. What we're going to shift to in this video and the next few are some methods that are less general, but in being much less general may be more particularly useful and helpful in solving the kind of problem we're faced with in protein design. Let's begin by switching over to the board where I can write. All right. So let's start off by just recapping the basic optimization problem that we're faced with here in protein design. We're trying to find a minimization, finding the best possible sequence out of all possible sequences. And we want to find the best energy, the sequence that has the best energy in the context of the target structure. But each sequence has a variable conformational space that we have to search over. And so that we might, inside this, place another minimization of XS over the conformational space of sequence S. And this other minimization is the energy of that conformation given the constraints of the target. And we can just kind of emphasize this has our constraints, our fixed backbone target. It has our continuous conformational search. And has our discrete search over sequences. Now, one of the things that we can, all of the methods that we're going to have share, all of the methods we're going to discuss moving forward share, is a reconfiguration of this problem into a single optimization rather than a nested optimization, right? And even though genetic algorithms allow us to interleave or combine the search to kind of perform these two minimizations and simultaneously, they're still treated fundamentally differently. We have sequence space, and then we have conformational space, and they're encoded in the description of the system differently. The structure of this problem turns out that we're able to transform this problem into a purely discrete problem with a few important approximations and understanding of the underlying system. The first that I want to piece of this puzzle is to really sort of think a little bit more about what these two optimizations are covering, the space that they're over. This discrete search is not just a discrete search. The variables are discrete. They're non-numeric. They're kind of just entities. But they're also finite. Right? And that means is that and typically this is about 20 per amino acid, which means this whole search space is generally 20 to the n total states, where n is the number of amino acids. Now, n might be big, and so we might have 50 or 100, and this might be a vast number of states, but it still is theoretically a finite search space. That is, there is a limit in the number of possible choices. Now, the optimization over the conformational space. This is, because it's a set of continuous degrees of freedom, there's an infinite number of choices. That is the set of all possible dihedral angles for even a single side chain. There's an infinite number of these because it's a continuous number. And a dihedral angle of 30 degrees is not the same as a dihedral of 30.1 degrees. And it's not the same as a dihedral of 30.12 degrees. Right? So we have this infinite number of choices per position, but, and this is a very important part, but a finite volume. Why do we say we have a finite volume? Well, they're continuous degrees of freedom over angular space. So each dihedral angle is limited to be on the domain of 0 to 360 degrees, or 0 to 2 pi if you prefer working in radians. Right? And what this means is that, yes, there's an infinite number of choices within this set, but the size of that domain is fixed, right? And actually, the volume of the space would be essentially 360 degrees to the n, or 2 pi to the n. And those are, once again, finite numbers, right? This just says basically by taking powers of the numbers of degrees of freedom, or the width of each domain. Now, these volumes don't mean anything. Right? I have no concept of what 2 pi to the n really sort of means, or what, but the important aspect is that they're finite overall volumes, and there's finite widths of every degree of freedom in the system. Why is this important? Well, this is important because a discretization or sampling of the domain leads to a finite number of discrete choices. Right? So what do we mean by a discretization? What we mean is that we can take something like the original, actually we don't even need to have this in 2 dimensions. So let me just If our original set of allowed choices is this interval that goes from 0 to 360 degrees of space. What do we mean by a discretization? It means that we replace that. We replace this continuously varying number line where every single point is allowed. We replace this with a finite number of points along that interval. Now, we can always discretize. We can discretize the set of all real numbers. We say instead of taking, allowing every possible real number, we're only going to allow integers, or we're only going to allow integer multiples of 5, or something like that. We can always apply a discretization. But if our volume of the search space is not finite, if some of the individual degrees of freedom are not of finite width, that is, they extend from 0 to infinity or from negative infinity to infinity, then when we do that sort of a discretization, when we do that sort of a sampling, we end up with an infinite number of choices, right? Or we have to simply cut off entire regions of the search space, entire volumes of the search space, which isn't necessarily valid. And so having a finite volume means that discretization can lead us to a finite number of discrete choices. Why is this particularly nice? Well, we started off the discrete portion of our search is involves a finite number of discrete choices. If we're able to also discretize the continuous space and move us into a finite number of discrete choices, what this means is that these now two entities, these two minimizations that we're working over, end up being exactly the same type of minimization, right? And what does this allow us to do? Well, sorry. So what this lets us do is then kind of to think about doing a little bit of a couple of transformations. And so we start with our minimization over sequences, our minimization over conformations of the energy of that sequence conformation given the backbone. Okay. And what we can replace this with, right? When we have, these are both the same type of entity, is replace this with simply a minimization over this sort of joint set of discrete possibilities of this energy given the backbone. And what the size of this search space will be dependent on how many discrete choices we have for each sequence. And so if we have m choices X of XS for each sequence S, or sorry, let's back up here. Sure. Then this would give us essentially 20 to the n. Actually, let me go back here. This is going to be confusing and it's going to complicate our later discussion. So let's just sort of back up and we will get into the discussion of how many states there are. All right. So this is our goal. Now let's think about what this discretization requires, right? And whether it's going to give us something feasible or not, right? So discretization is possible. Is discretization feasible, right? That is, does it make sense? Right? Now, the question here is going to come down to how many individual discrete states we end up with, right? So if we recall that we had 20 amino acids in general. And for each of these amino acids, we have 0 to 4 dihedral angles per amino acid. So let's just say on average, we have 2. Right? What does this really mean? Well, if we have 2 dihedral angles per amino acid, but we also have 20 different amino acids per position, we might expect that we have somewhere around 400 unique dihedrals per position. Right? If we had an average of 2 dihedral angles over 20 amino acids, 2 times 20 is 400. Right? Now, how many points do we need to choose per dihedral? If we sample, say, every 10 degrees, right? This means that the space 0 to 360 means that we end up with 36 unique choices per dihedral. Sorry for the spelling there. Per dihedral, which would lead to a search space of 400 choices times 36 choices per dihedral. So raised to the power of n, where n is the size of the system. Right? Now, this is a pretty big number. 400 times 35 is going to be somewhere or so times 36 is going to be somewhere on the order of 14,000, right? 12,000 from 30 times 400, and then another 2,000 from, say, 5 times 400, right? And 1,400 to the n, where n is maybe 50 or 100, is a really, really, really big number, right? If we have to sample every 1 degree, that increases that by 10 to the nth, because we have that much more sampling. This starts to seem like maybe it's not very feasible. But the underlying question is, how finely do we need to sample? And now here is where a very, very important experimental observation comes in. Okay, sorry. I'm trying to make an effort here to be a little bit more clear on writing. So there's a very, very important experimental observation. And this is that dihedral angles observed in proteins of known structure cluster. That is, they are far from uniformly distributed. Right? And so what do we mean by this? It means that if we take something like a one of the smallest amino acids that still has some degrees of freedom, something like, say, valine. Right? Valine has one dihedral angle. Sorry again. And if we plot just on 0 to 360, and we just look at every valine in the protein data bank, and we ask, what is the value of that dihedral angle? Well, what we see is that and this is made up data. So I actually do not recall, depends a little bit on which, where you start 0 from and things, but the rough idea is something like this, that you see that most of the results are clustered around a few of areas. And maybe every so often, there might be one outlier. Right? But the vast majority of cases are in these kind of wells, if you will. Right? Hard to see just points of data. And so we can create a histogram of this. And we can ask, what is the frequency as a function of the angle. And what we see in such a thing is that there may be a couple of small places where there's kind of one thing observed elsewhere, but what we mostly see is that there are well-defined regions where the vast majority of structures occur. Right? And we see that, so for example, we have three particularly sort of well-defined minima. And actually, this makes sense because we know actually that the energetic profile, even of just a dihedral angle by itself, is sinusoidal, right? And so we might expect the energy to have this kind of basic structure. And we see that there is a correspondence in the regions of low energy that we have higher populations. That's where we see things. Now, just an approximate dihedral energy function isn't always, doesn't tell the whole picture. There's more going on. We notice there's different heights, different sizes, different populations of these three. Some of these can actually be described because the dihedral energy, when we add up all of the different dihedrals, there's typically 9 on one atom, that they're not evenly weighted like this. And so perhaps a system like this might be better described by having one very deep minima, one less higher energy, and one medium energy basin to account for the different heights. And there's other energies involved, other things going on beyond the dihedral energy also. The key point, however, is that this is what we observe. And if we go to systems that have more than one dihedral, this pattern continues and can even be looked at in multiple dimensions. And so if we take a look, for example, let's say a residue like phenylalanine. Phenylalanine has 2 dihedral angles, and the same thing is observed if we plot on one dihedral angle from 0 to 360 and another dihedral angle from 0 to 360. Once again, we see a pattern of preferred states, that we see many observations in a few well-defined spots in space. And some of these may be very highly, oh, sorry, that shouldn't have been a line there. I'm not sure what happened there. But many of them may be very highly populated. Some of them may be very lightly populated, and some of them may be in between. But the most important feature here is that there is lots of empty space. And you could do a two-dimensional histogram to kind of highlight this also. Why does this matter? This matters because this helps us to have an informed discretization that does not have very many states. So perhaps we can choose a discretization. Choose a discretization that focuses our attention in these regions. So perhaps we discretize the valine dihedral space by choosing one choice at the peak of each of these choices. 3 choices, but where those 3 choices are right at the most commonly observed positions. And maybe in phenylalanine, we choose a set of choices that once again are centered at these most common choices. In the minimal set, maybe this is what we do, choose one particular choice per peak. If we wanted to have more sampling, if we thought we could afford more sampling, we may be able to do that by adding some extra structures in an informed way. So we might say, well, look, in valine, this central peak is very, very common, right? And there's significant populations off to the side. So maybe let's also sample a choice, a little sort of space away from that. So we have maybe 3 choices inside this region. And maybe we won't bother doing it for this one here because this is already a small population state. And so we can, in an informed way, add extra information to extra choices based on this distribution. Similarly, in the case of phenylalanine, maybe we say, let's add an additional choice at places nearby the center of these very highly populated peaks, but don't add extra information around some of the less extra choices around some of the less populated peaks. This idea that we can observe what are the most common structures and then define a set of choices that is based on those frequently observed choices is referred to as building a rotamer library. We use this term because each individual choice, each one of these individual structures, these preferred states, is referred to as a rotamer, right? And so we might say in our kind of simplest approximation, valine has 3 rotamers. Phenylalanine has 9 rotamers, and so on. Now, this is an approximation, right? It limits what we can find. we're trying to do a search over conformational space. And in providing this discretization, we are throwing out some possible solutions. For prediction, if we were trying to predict a protein structure, this might not be okay. Because yes, well, most structures, most dihedral angles for valine are in this scope. There are some outliers, right? What happens if our structure has one of these outliers and that outlier is important? The rest of the structure can't form without making that perturbation. It probably is the case, right? It requires energy to push that valine to a non-standard state. And maybe if we don't account for that, we will mispredict the structure. But design is a different beast. In much the same way as in designing an aircraft, we can say we don't want to consider all of the possible physics. We don't want to consider all of the different possible ways that wings can shape and be perturbed dynamically in flight. We can say we want to start with a very simple approximation that our wings are going to be fixed. And we're only going to sort of look at the problem within that constraint, and we're going to try to do the best we can within that constraint. We can do the same thing from design. We are going to say we want to find a solution that works with the commonly observed rotational states, rotamers. Yes, is it possible that you can make protein structures that have off-standard rotamers? Of course, right? But those are odd structures in some way. And by focusing our attention on only the best understood, the most represented side chains, we are making a simplifying restriction, but one that perhaps makes the problem more solvable. Right? Now, how big are rotamer libraries, right? What does this actually sort of lead to us? Well, for a simple case, right? Let's sort of just think about the simplest, the most kind of lowest or coarsest resolution sampling you can imagine. We can just kind of do a little sort of back of the envelope calculation of about how many states we might expect to see. Right? So what we're going to do here is we're just going to take a look at what are the possible amino acids. What is the amino acid name, the residue name? We're going to ask about how many dihedrals are there in this? And from this, we're going to estimate a number of rotamers. And we'll ask how many amino acids fall into that category, and thus how many entries in the library for that sort of class. There are two really big ones, lysine and arginine. These both have 4 dihedral angles. They roughly have, each dihedral angle has about 3 degrees of freedom, and this is a back of the envelope calculation. There are much more informed ways to do this, but kind of doesn't really matter for our purposes. We can say that, let's just say we assume 3 minima per rotatable bond. this gives us 3 to the 4, or 81 choices, different conformational states for each of those amino acids. And there's 2 of them. So this gives us 162 different possible rotamers just for those 2 amino acids. Then there's a whole set that have 3 dihedral angles. And I'm being a little bit loose here in sort of my definition of exactly what a dihedral angle is. Some of these have variations that actually don't map perfectly onto the classic 3 minima case, but again, back of the envelope calculations. What things have 3 rotatable bonds or 3 dihedral angles? Well, we have glutamate and its close cousin glutamine. We have methionine. We have tyrosine. And I'm going to include histidine in here because it doesn't have 3 rotatable bonds. It only has 2, but it has a bunch of weird protonation states that we also might want to include. So I'll throw it into this group. And this has 3 to the 3 choices. 3 choices per degree of freedom, 3 degrees of freedom. This is 27, but counting these up, we see we have 5 amino acids in this class. And so 5 times 27 is 135. Now we start going to actually the vast majority of amino acids have 2 choices per position. Serine, threonine, cysteine, aspartic acid, or aspartate, its close relative, asparagine, phenylalanine, tryptophan, leucine, isoleucine. These all have 2 dihedrals. 3 to the 2 is 9. And how many do we have here? 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 amino acids, but they only have 9 rotamers each. So even though this is the majority of, this is almost half of the amino acids that are possible, we have 81 choices only. Turns out there's actually only a single amino acid that has 1 rotatable bond, valine. 3 to the 1 is, of course, 3. Only 1 choice. This is 3 more. And there are 3 amino acids that basically don't have any flexibility. Again, I'm being a little bit loose in this statement for the case proline. Proline's a very weird amino acid that actually links back to the backbone. It does have a few different conformational states, but those are very, very tightly coupled with the backbone structure. When we have a fixed backbone structure, it is largely going to be the case that proline is fixed in its structure. And so we're going to consider these to be the cases where we have no dihedral angles. 3 to the 0. This doesn't mean that there's nothing we need to account. It means that there's 1 choice for each amino acid. There's 3 of this amino acids, so this is another 3 entries. And if we add all of these numbers up, we get our 20 total amino acids, and we end up with 384 choices. Okay. So approximately, we have a little under 400 choices in our minimal rotamer library. Now, actually, you see this even because of how the combinatorics works and how actually turns out that even though it's reasonable to say that kind of the average number of amino acids is or dihedral angles is 2. Our total space that we need to search is fairly modest. All right, significantly smaller than the 10-degree sampling, and really on par with what we had as our estimate if we had just asking how many kind of unique dihedral angles do we might think per position, right? Okay, so oh, sorry, this was actually, there's clearly some wrong math in here. I hope somebody had noticed this should not have been 400. This should have been 40. 2 times 20 is obviously not 400. And that changes this a little bit. Regardless, correcting that mistake, our overall size of our kind of minimal rotamer library, maybe 400. Much smaller, though, than having to sample every 10 degrees or something like that. Right? We can really get away with sampling roughly every 400 degrees, or every 3 times per 360, so maybe every 120 degrees. Now, what does this tell us? Right? That when we're now going to search over this, what are we really doing? We're searching over the possible choices of rotamers at each position. Once we've sort of put everything into this rotamer library, we're not even going to worry about what amino acid it is. We're just going to think we have 384 choices of different things at different positions, and this is how we're going to work forward. Now, what is then the search problem that we have, right? What we have is we have now a minimization over the set of sequences, but let's call these sequences sequence rotamer combinations. And we want to sort of think of this. We can sort of think of this as we have actually maybe some vector of S, where each SI is a rotamer at position I. And we want to then calculate the energy of this set of rotamers given our target backbone and find the smallest one. We have, for the smallest possible libraries, maybe 384 choices per position, and we have n positions. What this gives us is a total number of choices of about 384 to the n. It's worth exploring what kind of problem this is. So we've said that this discretization lets us move to an entirely discrete set, right? Where we can theoretically enumerate or count each possible choice. But how big is this, right? Well, for n equals 50, this gives us about 1.6 times 10 to the 129 choices. This is a huge, huge number. It's very hard even to sort of imagine how big these numbers are. And we get to big numbers, you understand maybe what a million is, what a trillion is. Do we even really understand? Do we really have a concept of that? And it's important to understand this because it's going to guide our choices and guide our thinking about how we tackle the problem. Just some reference numbers to help us think about what kind of number this is. Right? Very approximately, we may have something like 10 to the 80 atoms in the universe. What do I mean by that? If you sort of just think of the mass of the universe and convert that into number of hydrogen atoms dominate the mass. This is roughly the number you get. So this number is way bigger than the size of the universe measured in sort of individual atoms. There are again, approximately about 10 to the 60th or 10 to the 61 so-called Planck times since the Big Bang. What is a Planck time? A Planck time is the quantized unit of time. It's not something we really deal with very much because it is such a tiny amount of time that it's not something that we can measure. And so almost every event that happens is way beyond the Planck time scale. But quantum mechanics kind of tells us that everything ultimately has to be quantized. There's some lowest resolution that time can progress by that's kind of related to the Heisenberg uncertainty principle and so on. And if we define that Planck time and we measure since the dawn of the universe, since the Big Bang, we get only about 10 to the 61, right? So how big is this number? How big is this number of 1.6 times 10 to the 29th? That is the size of the search space for a small protein of only 50 amino acids with our coarsest imaginable rotamer library. Well, if we think about the product of these two atoms in the universe multiplied by Planck time, we get 10 to the 41. It's a little bit bigger than this number, but it's pretty comparable. So what we're saying is that for a small protein, the protein design problem, when we discretize it in this form, gives us a search space that's comparable to counting every atom of the universe every Planck time since the dawn of the universe. Right? This is a problem where enumeration isn't just hard. It's not just unfeasible with computational power as it exists. Enumerating through these choices and actually asking what is the energy of every one of these choices is something that will never be possible. Right? It would require all of the information in the universe to do that. Right? It's something we absolutely cannot do. Now, is this meant to show that the problem is unsolvable? No. Just like Levinthal's paradox in the protein folding problem that said that, look, if proteins had to fold by randomly exploring or systematically exploring every possible conformational state, that it would take billions of years for proteins to fold. The same thing is true here. Levinthal's paradox isn't really a paradox. It just tells us that's not how proteins fold. This limitation this beyond astronomical size of the search problem, simply tells us enumeration isn't a strategy, random sampling isn't a strategy. We need a strong bias in the problem. Okay. Now, in the case of protein folding, that bias, the solution to the protein folding problem, came from the structure of the energy landscape, these protein funnels. Those are evolved. Functional proteins evolved to have a funnel-like landscape that would allow them to fold in times orders and orders and orders of magnitude faster than that suggested by the Leventhal paradox. We don't know that sequence space, that this optimization problem has that same funnel-like structure. It kind of can't really have a funnel-like structure, because that even the idea requires ideas of distance, closeness to the structure, right? And so there has to be something else. You can still define sort of analogies to distance, and so maybe there's something of that structure there, but it's not something that's obviously apparent. It's not something that is necessarily has to be true. So what do we need to do? We need to have methods, algorithms that are able to solve this problem, this massive combinatorial problem, in a more effective manner. Now, although this massive search, the number of choices is huge, there is some light in the tunnel, or at the end of the tunnel. This is a very important observation, an important sort of piece of the puzzle for moving forward. And the point is the following, right? So this general problem that we're talking about is known as combinatorial optimization or combinatorial search. Given n positions with m choices per position, we get m to the n total possibilities. And it's this combinatorial exponential growth that is the huge, that gives us this massive search problem. Right? But that's looking at everything. Right? Now let's focus in at one position. But for a given position, each choice makes or can make, let me just erase that and write it more clearly. Each choice can make only n minus 1 times m unique pairwise interactions. where does this come from? Well, what we're saying here is if we think about we're thinking about, say, a protein, right? And if we think about all of the possible choices of all the rotamers in the protein at once, yes, we get this combinatorial problem. But if we ask, let's say I'm making one choice at a particular position here, and I ask, what are the ways that this choice can interact with other things? Well, there is say also a choice over here. And it might be it has some small amino acid choices. It has some larger amino acid choices that have a few positions. It has much larger amino acid choices that have more positions. And so this particular amino acid can make M different interactions with this particular position. But at some other position, there's that same set of choices. It can make a small one. There can be a big one that has a little bit of diversity in choices. And there are bigger ones that even have more diversity in choices. And there's M possible sets of those, right? If we've already focused our attention on one amino acid and one position, there's n-1 remaining positions. And so if we just add up all of these M choices at all of these n-1 different positions, these are how many individual pairwise interactions. Now, that was for the one choice at that position. If we now say, what are the set of all choices that we can make? And what are the possible interactions that one position can make with everything else? Right? Then what do we end up with? Well, we end up with M squared n-1 as the set of all pairwise interactions involving any choice at one given position. How do we get this? Well, we've said each choice can make n-1 times M pairwise interactions. We now add in the allowed diversity at this particular piece. We have M possible choices. Each one of these can make this n-1 times M pairwise interactions, and we get M squared times n-1. Now, that's the interactions that one position can make. If we now ask, what if we look at all possible positions? And we ask the same question, right? What are the pairwise interactions that we get out of this? Well, we get the following. We now have to take this basic number and multiply it by n. N times M squared times n-1. And there's going to be one modification that we make in a minute, as the set of all pairwise interactions But if we just do this, we've actually done some double counting, because for each of these choices, right, we've already counted. When we get over to now to this group here, well, we've already counted the interactions that we made over here. And so we need to really divide this number through by 2 to give us our actual number of pairwise interactions. Now, this is the set. So this number tells us how many unique ways can I choose a pair of rotamers at any position with any of the rotamer choices, but limiting ourselves to a pair. Now, if we throw in our approximate our small system, the numbers for our small system of say n equals 50 and m equals 384, this gives us approximately actually gives us approximately 2 times 10 to the 8th pairwise interactions. Now, 2 times 10 to the 8th is 200 million. 200 million is a big number, but 200 million is not an astronomical number. We deal with numbers of that size all the time in computational work. Just think about how we measure memory and processor speed. We measure memory in the megabytes or gigabytes. We measure processor speeds in gigahertz. That's megabytes are millions of bytes. Gigabytes are trillions of bytes. Gigahertz are trillions of cycles per second, right? Billions of cycles per second. So we regularly work with numbers in the millions, billions in computational problems. So this is big, probably expensive to calculate, but not prohibitively so, not out of the realm of possibility. Now, why is this okay? Why is the fact that pairwise interactions are suitable? Well, it turns out that the underlying molecular mechanics energy function, right? And if we just think about this, what is the molecular mechanics energy function? It is written as a, if we kind of take it in the standard form, we take a sum over all of the bonds and a bond energy, and a sum over all of the angles, and have an angle energy, plus a sum over the dihedral angles, and then we add to this a sum over all pairs of a Leonard-Jones or a van der Waals, plus a Coulombic or an electrostatic energy. If we notice this piece here, the non-bonded interaction, this is written in terms of pairs of atoms. The non-bonded interaction is a pairwise interaction in terms of atoms. These are a little trickier. Right? The bond term does involve pairs of atoms. The angle term involves triples of atoms. The dihedral angle term involves quadruples of atoms. But we also have to remember protein structure and the fact that a protein has a backbone, and in protein design, the backbone is typically fixed. And so let's think about this a little bit. Let's start with a alpha carbon. And we have a carboxyl group. And so we have some side chain, and let me call this R1, or I'll do it R-1 for ease. And then we have our amino group, and we have another carbon, and we have our side chain I. And we have our side chain I+1. The important thing to note about this is that if we count, so remember, in protein design, our backbone piece, actually we should kind of include these. Our backbone piece is often fixed. It's part of our constraints, it's our target, right? So this part isn't variable. This part is something that we know, it's a given of the problem. Now, if we just count bonds, we can count here. We can say if I go from this position, right, it takes one bond to get to the backbone. Takes another bond to get over to the carbon. It takes a third bond to get to the nitrogen. It takes a fourth bond to get over to this carbon. And it takes a fifth bond to get over to the side chain. What does this mean? This means that the side chain atoms of one amino acid and the side chain atoms of another are always separated by at least 5 bonds. Why does this matter? Well, these bond terms are interactions between atoms that are connected by one bond. They're so-called 1-2 interactions. Angle terms are interactions between atoms that are connected by 2 bonds, 1-3 interactions. Dihedral angles are 1-4 interactions. They're interactions that involve interactions through 3 bonds. What this means, is that the limit of the bonded interactions. So if we think about taking this residue Ri, and we say, how far do the bonded terms reach? Well, the bonded terms can reach as far as 1, 2, 3, 1, 2, 3, 1, 2, 3, and 1, 2, 3. So we kind of have a regime of where the bonded interactions of a side chain can reach, and they are still separated from the other side chains. Side chain interactions are non-bonded interactions only. Non-bonded interactions are pairwise interactions. The interactions that a residue makes with the backbone might include the bonded interactions, but that doesn't matter because the backbone is constrained. The fact that side chain interactions are non-bonded and side chains are and non-bonded interactions are pairwise decomposable tells us that the overall protein design problem can be broken down into a set of pairwise interactions. That's where this fact that we can compute all of those pairwise interactions comes into play. All right. At this point, we have gone through, introduced the idea of discretizing protein space, developing the a rotamer library to describe the actually quite limited number of rotameric choices that are reasonable choices at any particular position, and how, although the combinatorial search problem for this totally discretized problem is beyond astronomical, we can afford to compute all possible pairs of interactions. And something about the structure of the molecular mechanics energy function suggests that this pairwise decomposability is built in to the energetics that we need to calculate. In the next video, we are going to expand on this a little bit more, clearly defining a pairwise decomposable energy function that can be used for protein design, and then introduce one of the algorithms that we can use to tackle problems within that framework. In subsequent videos, we'll explore a few more algorithms that use that same energetic and rotamer library structural framework for solving the problem. Until then, take care and be well.